our next presentation, I will introduce Dr. Kathrin Meyer, who studied cellular and molecular biology at the Institute of Cell Biology in Bern, Switzerland. Her postdoctoral research was performed in Brian Casper's laboratory at the Center for Gene Therapy in Columbus, Ohio. During that time, Dr. Meyer established a new and fast reprogramming method for in vitro remodeling of neurodegenerative diseases using patient skin cells. Moreover, Dr. Meyer developed intrathecal gene therapy programs for several neurodegenerative diseases, including spinal muscular atrophy and Batten disease. Multiple clinical trials that are based on this work are currently ongoing at Nationwide Children's Hospital. In 2017, Dr. Meyer became a principal investigator at Nationwide Children's Hospital. She is also an assistant professor in the Department of Pediatrics at The Ohio State University in Columbus, Ohio. Thank you for joining us today, Dr. Meyer. I will hand it over to you.
Thank you very much for the kind introduction. Can you hear me all right?
I can, yes, thank you.
Wonderful. All right, let me share my screen. All right. Okay. All right, hello everyone. I am really excited to be here today. Here you see a picture of our patient hospital wing at Nationwide Children's Hospital. The research building is right behind that. These are my disclosures. And today I'm going to talk about a couple of different things. I've decided to first start with the clinical trials and give some thoughts and considerations around clinical trials from a researcher's perspective. So I'm not an MD, I'm a PhD, but I think some of the thought processes might be interesting. I'm going to talk about ongoing research and also challenges in translating research to the clinic, and then add some future directions, areas that I think could be really interesting to further improve therapies or develop therapies for spinal muscular atrophy. And then I'm going to also briefly at the end talk about a gene therapy that we're currently developing for a different form of SMA, spinal muscular atrophy with respiratory distress type 1. So when you look at the landscape, the type of treatments that we're thinking about in spinal muscular atrophy, and I know you've been talking about multidisciplinary care earlier today, and I think also after me there is going to be a talk around that. That's really the center of the piece also for the clinical development and for research. There are SMN-dependent pathways, so pathways that we're trying to target that will influence the expression levels of the survival motor neuron protein. So we can make more protein, or we can activate the background, the backup copy of the gene, the SMN2 transgene. You've heard about splicing today. There's also approaches that allow stabilization of the protein, so the degradation of the smaller amount of protein that is there will be delayed. But then there's also independent pathways that don't target directly the SMN protein. So there are approaches that are trying to work on cell therapies, stem cell transplantation, for example, neuroprotection to make the nerves that still exist, the neurons, stronger. There are therapies that are looking at enhancing the neuromuscular functions, so the muscle-neuron interaction, and ultimately also therapies, a couple of therapies that are looking at muscle-enhancing performance. So really all of these have a different function that are potentially promising or interesting looking at SMA research. This is a table that tells you a little bit about different types of clinical trials. For example, here the SMN-dependent group. So here we're looking at alternative splicing. You've heard of nusinersen, risdiplam that are now FDA approved. So those are really, really promising approaches. Then of course we have the gene replacement, Zolgensma, but there's also other small molecules that have been tried in order to upregulate SMN expression, including BPA, HDAC inhibitors, etc. And then also down here, there are some that can stabilize SMN protein, the SMN protein itself. All of those are still in preclinical development. Independent axis of clinical trials or therapies that we're trying to develop The muscle enhancing agents have recently shown promise. There is the SRK-015 that's in ongoing phase 2 open label, and a couple of other promising targets. So they're mainly targeting myostatin, inhibiting a muscle inhibitor so that the muscles can grow faster and become larger. And then also neuromuscular junction defects, as I mentioned, or neuroprotective agents as well. So when you look at the SMN-dependent strategies, as you know, three treatments are currently approved: risdiplam, nusinersen, and Zolgensma as well. Zolgensma for intrathecal delivery was put on hold in 2019 due to potential DRG toxicity pathology that was found in non-human primates, but the hold was recently lifted in 2021. Currently, the intrathecal delivery is actually restarting in a pivotal trial that has recently been posted on clinicaltrials.gov. This will be a multi-center, randomized, sham-controlled phase 3 clinical trials for patients between 2 years up to 18 years of age. And at the time the clinical trial will finish, patients that were in the sham control group will be able to roll over into the treatment group as well. These will be naive type 2 SMA patients with 2 to 4 copies of SMN, meaning they will be able to sit, but they were never ambulatory. Okay. The myostatin inhibitor, as I've previously mentioned from Scholar Rock, also showed really positive results in patients. Those have been recently published. And what's interesting is that the results were particularly good in combination with the nusinersen treatment. You can read that up further online. The clinical trial is called TOPAZ, and there were some really interesting results there as well. I guess in general, when you look at all these clinical trials, including the myostatin inhibitors, nusinersen, risdiplam, Zolgensma, I think one of the common themes that we see is that earlier treatment is better in almost all cases. It's just much easier to prevent neuronal loss and muscle atrophy than trying to revive cells that have been severely damaged or cells that are already dead. I mean, you can't revive something that's not there anymore. One of the other things that's also important is that there is a significant variability in the patient response. So there are patient subpopulations that show a stronger or less strong reaction, even if they were quite similar at the time of treatment. And that is something that we don't fully understand at the moment. Combination therapies might have additional benefits. I think there is still a lot that we need to learn about it. So we don't have complete answers there yet. We don't know which combinations will be best for which patient, for example, and which order of treatment would be best. Is it better to take nusinersen first, after risdiplam, Zolgensma, et cetera, et cetera? So all of that still has to be evaluated. There are several clinical trials that are currently testing combination treatments. And obviously treatment options have changed for spinal muscular atrophy because of these life-changing therapies, but also because of all the clinical trials that are ongoing. The standard of care has also changed for the patients, and recent guidelines have been released. And I think really the combination of these two aspects is what helps further develop and improve the SMA treatments for the patients. So in terms of combination treatments, as I mentioned, there is a clinical trial ongoing with nusinersen and risdiplam. This one hasn't yet started recruiting, but has recently been posted. There is a treatment testing going on between nusinersen and SRK-015. This trial is active. It is not recruiting anymore. There is a combination therapy that's currently not yet recruiting, but will be soon, between risdiplam and the myostatin inhibitor from Roche. And then also an ongoing recruiting clinical trial that is evaluating nusinersen and Onasemnogene Aboparvovec, so Zolgensma gene therapy in combination. So ultimately, why are we doing all these combination treatments? Obviously, it is because we're not yet completely satisfied. We don't have a cure yet. And also, when you look at the SMA disease, and as you obviously heard probably earlier today, it is a disease that affects multiple organs. It affects the brain, it affects the heart, the muscles, bone, intestinal tracts. And for some of these organs, we're actually not sure, because sometimes what we see in an animal model doesn't necessarily translate to a human being. So when you look at some of these images here, certain observations that we made in the mouse haven't yet been shown in human patients. So there's always a little bit of that. The clinical data is really important for us to see. The ideal treatment, of course, for SMA would be a treatment that affects multiple organs throughout the body, because if one thing is clear, it is a multi-organ disorder. It affects the entire body. The organ systems that are affected are tightly connected with each other. So neurons and muscles, of course, as you know, they are interconnected. They work with each other. So if the neuron is sick, the muscle is going to suffer, and vice versa. If the muscle is sick, the neuron will suffer. That's obviously a very obvious connection. But when you look at the gut, there is something that's called the gut-brain axis, which means that the health of the gut can also influence, in particular, inflammation and health of the nervous system. And the same is true for metabolic aspects, and of course, blood vessel integrity, et cetera, et cetera. So what's currently unclear is what happens if you fix one department to a very reasonable extent, but not the others. Are other elements going to fall in place, or do we have to fix everything simultaneously? And that is very challenging because, as you know, the body is really large. Each organ has different requirements, different barriers of blood, drug uptake, et cetera, et cetera. So, and the second question is, what can we model and what can we not model in animal models, basically? I really like this graphic from a review from Dr. Tizzano and Dr. Finkel from 2017. Another aspect that's going to become really important now in the treatment of the patients is that the phenotype, so the disease presentation is changing over time. So an SMA type 1 patient might, that initially would have had very severe, very fast muscle loss and muscle atrophy, would have been paralyzed or would have unfortunately passed away within the first 2 years of age, well, has now several different options on how this disease can go. Either, as you see on the bottom here, we could see a stabilization with a better function of the remaining muscles and nerves, or we could see an improvement, muscle growth, movement, with maybe patients being able to sit, or we could have patients that are walking, et cetera, all the way to almost a near cure. And what that means, which I really want to point out, is that this is actually quite difficult for the physicians, in my opinion. As I said, I'm looking at this from the standpoint of a researcher, because the physicians now have to really relearn what SMA means. The presentations are very different, and they might see symptoms that they haven't seen before because the patients live longer, because the patients are stronger and they're able to do things that they weren't able to do before. And these kind of observations are going to be very important for us researchers so that we can address whatever comes up next moving forward. And as the experience with these new therapeutic increases, we'll also be able to better adjust the treatments for the individual patients. I think at the moment, what I see, or when I also talked a little bit before this symposium with my friends and colleagues that are in the clinic seeing the patients. I think there is a little bit of not frustration, but confusion, because there are so many different options. And the question is, how do you choose the best option for your patient? And that is a question that I think we haven't answered at this point. This is a good chart as well that kind of addresses the same question. How do we treat the SMA patients? So depending on the different therapeutics that we develop, the first question is, okay, who should we treat with this? Should it be for all SMA patients? Should it be for patients that are younger, less affected, more affected, older? Should it be pre-symptomatic? What are we going to treat? Are we going to treat the gene defect, or are we going to treat an SMN-independent pathway or a combination? Now, where do we apply this treatment into? Are we putting it into the spinal fluid? Are we putting it into the bloodstream? Are we putting it orally? And how does it get to where it needs to go? And then the therapeutic window, as I mentioned, when do we treat the patients, and how often are we treating the patients? So these seem to be quite simple things when we're just talking about, but I expect to see many more clinical trials that are actually going to be tweaking some of these aspects. So just because we have a certain treatment in the clinic now doesn't mean that this is perfect. We can still improve delivery, we can still improve dosing, and we can still potentially improve the patient subpopulation that can best respond to the treatment. So all of these things go into research, both on the preclinical side and on the clinical side. All right, so I'm going to talk a little bit about some of the research that we've been working on, and some of the research that recently has been published that I think underlines some of the challenges, but also promises in multiple areas. And it's going to be a little bit focused on gene therapy, because that is, of course, my main area of work. But a lot of the things that I'm going to talk about are going to be applicable for multiple disease and drug development studies as well. So a major challenge that we're facing on the preclinical side is the dosing and delivery. So it's really hard to go from something that is as small as a mouse that fits on your hand to a human patient. And oftentimes we use, as you can see here, non-human primates as a sort of a bridging species that is a closer relative to humans, but also larger. We also sometimes use pigs, but ultimately we need to find a way to kind of scale up from the mouse to larger animals. The problem that comes with that is that the nervous system is also not built the same way. So the number of neurons or nerve cells in a mouse don't scale up linearly to a non-human primate or to humans. There's also different composition of cells in the nervous system, different volumes, et cetera, et cetera. The permeability of the blood-brain barrier can also be different between the species, and also how that permeability changes at certain ages is different. And then ultimately, something that I think a lot of people oftentimes forget is also the drug uptake. So there can be metabolic differences between the animal species, but also even just the surface receptors, for example, that we need for uptake of viral vectors can be different. So another aspect in the preclinical work that is challenging is when we work with mouse models or mouse species, they are what we call inbred. So they have been designed to be genetically very similar to each other, so that we can sort out some of the noise that we see in a normal population, which is great because it allows us to cleanly see certain pathways. But it's also not great because that is not what happens in the clinic. That is not what happens in patients. Humans are not that equal. They are very different genetically, and there are modifiers that we don't understand at this point. So while all of the research I talked about is very important, mice, non-human primates, humans, I think that the most important part is really learning from the clinic. It's a little bit unfortunate in a way because we want to give the best treatment to the patients as with the first trial if we can, but unfortunately there's a large learning curve in the clinical trials. But that's also what makes me really hopeful for SMA, and I think the development will continue to accelerate because now we're having all these therapies in the clinic that we can learn from. Here's an example that kind of underlines this. This is recent research trying to improve viral vectors. So how can we make viruses that are more efficient in getting into the nervous system and targeting the cells in the nervous system so that we potentially can reduce the dose, which could make the treatment potentially cheaper, but also would make it more safe. And what you look at here is a comparison between the AAV9, which is currently used in Zolgensma, and the new vector surface, so the protein structure around the gene therapy, that obviously looks much better. So every green cell here has taken up the virus. So when you just look at this, you'd be very excited and say, yeah, that's exactly what we need. But unfortunately, this vector only worked in a certain mouse strain and not in another. And so when you think about, when you go back to that picture of all the different patients, you can actually imagine that a situation like this could very much apply in human patients as well, and could explain why some of the patients might react much better to the gene therapy than others. So the larger animals allow us to hopefully rule out some of these things, but they're not going to be a complete model for that. And it comes to a second question, especially in gene therapy, but I would say in general also for other therapeutics for the nervous system. How do we know that we're actually targeting the right amount of cells and have the right amount of kinetics in the nervous system? So we can look at that in non-human primates, but oftentimes we don't have data or information from human patients. This is really an important aspect for all treatments. And so I wanted to highlight this research paper that has recently been published by Dr. Mendel and Dr. Faust and the group of Novartis around the biodistribution of the gene therapy in human patients. Unfortunately, there were 2 patients that died in the phase 3 clinical trial. The deaths were not related to the treatment, but they were related to respiratory complications. And while that's very sad, this was really good for research because it allowed us to finally look at the distribution in human patients that have been treated. So that we can understand, is the current dosing actually giving us the optimal treatment, or is there room for improvement, or are we too high? Can we reduce the dose? And so basically what the researchers did here is they evaluated the distribution of the gene therapy vector, as well as the expression levels of SMN, both on the RNA level and the protein level. All right, so here you can see in both patients side by side the distribution of what we call viral genomes. So how many copies of the vector can you find in different organs? Over here you have the spinal cord, brain areas, muscles, and then also internal organs as you have here, heart, lymph nodes, spleen, and then the liver. And the way we're measuring this is a calculation where we're looking at the viral genome copy numbers per cell, basically. So how many copies do we have on average per cell? And you can see that in most cases, the average copy was between or around 1. So this is pretty much what we would like. However, when you look at the liver, the copy number was between 300 to 1,000-fold higher than what you see in the CNS. So basically, and we know that from preclinical work already, when the gene therapy vectors are delivered intravenously, a lot of it ends up in the liver. And that causes, toxicity, and this is really a rate limiting and a dose limiting implication there. So as a next step, the researchers took the spinal cord, and we have ways where we can actually cut out the spinal motor neurons from the spinal cord and specifically look at the expression levels only in the motor neurons themselves. So here you can see the vector genome copies in the motor neurons in these 2 patients. And you can see that you had about 1 to 2 vector copies per cell on average per motor neuron. And when they looked at expression levels now and compared it to the expression levels of SMN of a healthy individual, the expression levels were actually still pretty low. They were around 25%, 25 to 30% of what they saw in a healthy control individual. So what that would tell us is the expression levels are still below healthy controls. Now, it's not clear if that will be the same in all patients, but at least in these 2 patients, that was the case. So this could indicate that there is further room for improvement of the gene therapy. They also looked at a distribution of the vector in the peripheries, looking at the muscles and other organs, and they saw really good distribution. Everything that's dark brown is SMN protein expression. This is compared to a non-treated SMA patient, so you can clearly see a very strong difference there throughout all the tissues looked at. So the vector copies were correlating very well with the expression levels seen in the different tissues. So the expression in the periphery seemed to be fairly strong as well. So as I mentioned before, there are a couple of good points here from a research standpoint. The biodistribution was similar to what we saw in non-human primates, so that's good. The widespread targeting of the CNS and the periphery shows that with the gene therapy, we can actually target pretty much the entire body, or large parts of the body at least, so that's also good. However, as previously been afraid of and seen from some of the reactions and preclinical studies, the liver is very highly targeted, and the SMN expression is still lower in the CNS compared to healthy controls, so there will be room for improvement if we could go higher with the dose. In terms of a higher dose, there was a recent publication that was quite interesting that was talking about potential concerns when overexpressing SMN. So this is a study from Dr. Pellizzoni. It's really nice work from his group. And what they did basically is they used the same AAV9 vector serotype, so the same virus as we use for Zolgensma, to deliver the SMN transgene. But you know, the vector production was different. Of course, it wasn't the same as what we use in the clinic. And then also they used a different promoter compared to what we used in Zolgensma. So the promoter they were using is supposed to give actually a lower expression compared to what's used in the clinic right now. What they found is that the construct was working really well in SMA mice, just as previously described. But when they treated wild-type animals, so animals that already have 2 healthy copies of the gene, they started to see that they got clasping phenotypes, which you can see here in F, and it's quantified over here. They also saw a reduced weight gain, a reduced survival, and a loss of strength of these animals, so post-treatment. In addition to that, they found when they sacrificed the animals and looked at the spinal cord and other the peripheral neurons, they saw that the SMN protein was actually aggregating. So here in green, you see those protein clumps. That's actually a staining for SMN. And so you see that you have aggregation, and that's quantified over here in the graph right there. The aggregates were only seen when the animals were treated with SMN, not when they were treated with a control virus that contained a different protein. In addition to that, they found that when they followed the mice for 300 days, which is pretty old age for mice, they found degradation of neurons both in the dorsal root ganglia and also in the spinal cord. They saw reduced numbers of motor neurons, which you see down here in E, and also reduced size of the motor neurons. There was a pretty clear effect of this treatment. Some of the effects they already saw early on, and some of the effects they saw at later time points. Now, it's a very elegant study. It's unclear, however, how this translates to the clinic. Again, looking at a mouse as a mouse model versus larger animal models, where I don't think any of this has been described, versus humans, because we usually tend to overdose mice. We give them a larger dose per number of cells compared to what we do in humans. There are also certain differences in the vector construction, as I told you, production, et cetera, that can make a pretty big difference and make it difficult sometimes to compare studies between each other as well, between the researchers. In addition to that, in clinic, we're actually treating patients that have reduced levels of SMN, not healthy patients that already have normal levels of SMN. So all of that, of course, makes the interpretation or the thoughts towards clinic difficult. However, I think it is still something, this study still tells us something that is worth, definitely worth listening to. Those are considerations that we should keep in mind for type 2, 3 SMA patients that already have higher levels of SMA. Also, for example, I just showed you the liver is highly targeted, so there could be other organs where overexpression problems could become a bigger issue moving forward. And then there's also the fact, for example, that the dorsal root ganglia, which were clearly affected here in this study, are highly targeted by AAV vectors. So clearly something that we need to keep in mind moving forward, and when we design potentially new second generation treatments, we should be wary about that as well. So in order to try and reduce the exposure of the periphery, one of the aspects that one can use is, of course, instead of putting the drug into the blood flow, we can put it directly into the spinal fluid. That will allow us to circumvent some of those overexpression issues. We can target the peripheral organs. We also don't have to dose per body weight because we have direct access to the nervous system, and the differences in the size of the nervous system is not as big between patients compared to the weight. So you can use much lower doses to reach the brain and spinal cord. And, you know, for example, nusinersen takes advantage of the CSF delivery to get to the cells more efficiently in the nervous system. So we're avoiding to have to find our way all the way in there. So we did a study comparing intravenous delivery and CSF delivery side by side with the exact same viral vector construct in a similar disease that's called amyotrophic lateral sclerosis, so a motor neuron disease for adults. And what we found in this study in mice is that compared to untreated animals, which you see here, they die around 130 days of age, the intravenous and the intrathecal delivery were absolutely equally efficient, but with the intra-CSF delivery, we were able to use a 10-fold lower dose. So that per se obviously adds a lot of safety right there. However, Dr. Wilson's lab has also recently published several studies looking at the dorsal root ganglia, and with the CSF delivery as well as with the intravenous delivery, the DRGs are highly targeted. The good news is though that despite the targeting, the pathology that is seen in the dorsal root ganglia throughout multiple studies with hundreds of animals that they compared was very mild or minimal. There were no clinical symptoms in non-human primates that received therapeutic transgenes, and this includes 33 studies and over 250 non-human primates. So this is a really important step. So oftentimes researchers use what we call reporter genes, so green fluorescent proteins or other reporters for us to better follow the delivery aspect of our therapies. And those can be toxic by themselves. It's really good news, even though we targeted DRGs at very high levels, at least in non-human primates, there has been very minimal to no pathology throughout. There are 2 clinical trials with CSF delivery that have seen a DRG-related pathology, one in ALS and one in an autopsy of a patient in the GAN trial as well. Other than that, to the best of my knowledge, from what's published, there haven't been any issues with potential pathology in the DRGs in human patients either. The DRG pathology could be impacted by many things. The dose, of course, the promoter, so how strong the expression level is, delivery routes, but then also what type of immunosuppressive protocols are used when we go into the clinic. And so this is basically just a little summary or a reminder that when we use the CSF to deliver drugs, any drugs really, there are different kinds of entry points that we can use. So we can either go through the brain and reach the intracerebral ventricular area and then inject the drug directly into that space, into the CSF. or cisternomagna injection, which is right here at the just at the bottom of your head, or a lumbar intrathecal injection, which is the most commonly used and probably the safest or easiest procedure, let's put it that way, that can be done. So all of these different routes are currently in clinical trials for multiple types of therapies, be it ASOs, small molecules, or gene therapy as well. The problem with all of these delivery routes at the moment is that the distribution throughout the nervous system is still not even. No matter where you access the spinal fluid actually circulates throughout your brain and spinal cord, but you don't get an even distribution. So a lot of research right now, as well as clinical trials, are going into how can we improve the delivery. And basically, to also try and avoid, which you heard just in the previous presentation, to avoid having to get repeated access to the CSF. As you know, nusinersen has to be re-injected into the spine and into the CSF at multiple time points, and therefore repeated access is needed. So there is research that goes into potentially developing pumps or ports that allow easier access both into the ICV injections, lumbar intrathecal, or for ICM, I don't think there is any ports right now. So that's a big area of research. The current challenge really that isn't solved even with being able to create better access to these injection routes is the distribution throughout the nervous system. So how do we avoid hotspots? Because we just discussed they can potentially be dangerous. This is just a little illustration. This is data from a non-human primate that we had dosed lumbar intrathecally, and we actually tilted the animal with head downwards to improve the spread towards the brain. And as you can see, we had good distribution throughout the brain. So these are just controls over here. But this is really like a valley mountain kind of situation where you have a lot of virus in certain areas, and then especially in the deeper brain structure, you have much less. And similar, distribution issues can be seen with other therapeutics as well. Each therapeutic, depending on, the size of the molecule and the formulation, has different advantages in terms of how it spreads throughout the nervous system. So what can influence distribution? It's of course the dose. If you put more into the nervous system and you have a reasonable way to spread it, you will probably get more cells targeted. But then, as I mentioned, the formulation, what kind of material is your drug in? Is it a water solution? Is it a fatty solution, et cetera, et cetera? The injection volume can play a role, as well as specific injection procedures, needles, the orientation of the patient, or pumps that can be developed to better spread therapeutics. There are also other factors that you're probably well aware of, such as scoliosis, which can make it more difficult for the therapeutics to actually spread through the CSF. So all of that, again, adds to the diversity and to the potential differences in the treatment response that we see. So there could be, aspects just from the patient's morphology that would warrant the investigation of a different type of injection route or a different type of injection level, just based on the disease progression of the patient. I'm just going to show a few examples of recent publications that talk about combination therapies. So we're obviously evaluating, the research community evaluate these combination therapies in mouse models as well. This is a study by Dr. Burgess, where he combined the antisense oligonucleotide and another splice modulating drug. And he dosed the mice at different time points with the antisense oligonucleotide, either at 2, 4, or 6 days, and then continued oral treatment with the other splice agent. And what he saw, of course, is that especially for patients that were dosed at a later time point with the antisense oligonucleotide, there was a really big difference in the response to the treatment with the combination treatment. So we really think combination treatments are going to be very, very important moving forward. This is another study that has been published recently where a combination treatment of valproic acid and the morpholino splice switching oligonucleotide was studied. This was done in patient fibroblasts, which is something that I just quickly wanted to point out here, and it's going to be important for the next part of the presentation that I'm going to talk about. And then another area of interest, of course, that I'm not going to really talk about too much today is cell transplantation. There have been trials to use stem cells of different kinds, either human-induced pluripotent stem cells, patient-derived stem cells, or mesenchymal stem cells that can be injected into the spinal cord at different areas. They can provide growth factors, neuroprotective support, but unfortunately, the idea or the hope would be that these cells could grow into motor neurons and could actually take over some of the work that the lost motor neurons can't do anymore. This is still very challenging at the moment because the problem is when we're putting these cells into the spinal cord, first of all, they don't really usually travel very far, so they stick just around where we put them in. And then also they lack the cues to properly mature into the neurons that we actually want. And then also they lack the cues to actually grow out into the periphery and then make the proper connections with the muscles. So there's still a lot of work that needs to be done there. Some interesting efforts, of course, are coming from the field of spinal cord injury as well. How can we get neurons to grow back out from the spinal cord? But it's a very challenging field. The alternative that I think is quite interesting, in fact, but also still very futuristic, is instead of transplanting new cells into the spinal cord or into the brain, the researchers are currently developing viral vectors and other strategies on how they can potentially convert other cells that are not neurons in the spinal cord in living animals to become neurons basically on the spot. So there are support cells that we call astrocytes that are very abundant in the spinal cord and the brain, and they can actually divide. So this is a source of cells that can be renewed. And there are multiple efforts by many research groups trying to see whether we can turn these astrocytes into neurons. And there are a few recent papers that actually show that with these kind of conversions, you can not only rescue and generate neurons in the right spot, but you can have a functional improvement. Both here, this is an example after an ischemic stroke, where animals treated with the gene therapy vector were doing better, both on like a Rotherow test, but also were faster in swimming, which you can see down here. There are also tests going on in Parkinson's, for example, with similar kind of results. So this is still very futuristic, but that could be in the future an additional combination treatment that could become interesting in SMA should we ever manage to get motor neurons to grow back out. Of course, the next question right there is then, are the muscles still going to be able to respond? Are still additional muscles going to be present that we can reactivate? But muscles, lucky enough, they have stem cells, so we could potentially, with regrowing the muscles, there might be some futuristic ideas there where we can get with these kind of research, but this is going to still take a pretty long time. So the last part of the research I want to talk to real quick is about the use of in vitro models to study the disease mechanism and to evaluate therapeutic strategies. Why do we need in vitro model? In vitro model means we're using tissue cells from the patients, either isolated from the blood or from the skin. We can use these to really understand patient subpopulations and try to predict responses to a given therapy, which is what the MDs are really struggling with right now. But then also, some of the things that we see is that the genetic reports don't always predict the effect of the mutation. sometimes the cells manage to do something very different with a mutation than what is predicted by the genetic report. And then also it will potentially help to improve clinical trial design, inclusion, exclusion criteria, et cetera, et cetera. Ultimately, we can study diseases that have no animal models with such methods as well, which is important, especially for some of the more rare forms of SMA, where we don't have as many model options compared to SMN-caused SMA. All right, so I'm going to go a little bit fast here because I think I'm running out of time. This is just an example of how skin cells in a dish look like. And then when we change them into neuronal cells or neuronal progenitors, how they can completely transform in something different. I'm going to give you one example of where we've used this technology on ALS. We've actually made a specific subtype of cells here, which are called the astrocytes, as I told you, and we looked at how the astrocytes impair the neuronal function. And the reason why I'm bringing this example here is that we found a potential therapeutic, which is a small molecule, it's called copper ATSM, that is currently in clinical trials, not from us, so we didn't find it, that has a really strong effect on the impact from the astrocytes. But what we found when we tested different cell lines from ALS patients is that there were some patient cell lines that did not show a response at all. And that's really something that we need to understand. So that patient in the clinical trial is not going to benefit, but is also then going to reduce the power of the results. We then could further analyze what is the molecular mechanism that differentiates the responders from the non-responders. And in this case, it was had to do with the energy production. So this is really something very important to keep in mind, where we can help the MDs, hopefully in the future, to better stratify their patient populations. Then also, one of the things we can do is use that knowledge and test it very quickly on other diseases. Here is an example of a loss of function mutation in a sodium channel. So on the genetics, on the paper, the diagnosis was loss of function. But when we did a sodium uptake assay, this loss of function mutation actually had a higher sodium uptake compared to other cell lines. So clearly, the cells found a way around this, and that's a very different disease mechanism right there. So we've got to be careful with that. Ultimately, this has already been, done to some extent in SMA with the response to VPA. So there are publications that show that certain patients respond well to VPA, certain SMA patients don't. If we could just figure that out with the patient cell lines, that could really, really help, and it would reduce the burden of the patients to go through multiple clinical trials that might not even be right for them. So it really goes into that personalized medicine aspect. And that's what we're really interested in. And also, that's why it's so important to participate in research. And we're really grateful for all the patients and families that, donate tissues and help with research. It's very, very precious. So yeah, that's kind of the summary of the in vitro modeling part. It will help us to get away from this one size fits all. It might help us to discuss with the MDs what would be the best approach to move forward for individual patients. And then I'm just going to mention this very quickly. As I said, we're developing a AAV9 gene therapy for spinal muscular atrophy with respiratory distress. This is a very severe disease that leads to respiratory failure within 6 months of post birth. And then patients are ventilator bound for the rest of their life. There are milder forms of this disease as well. So it's a very similar situation to SMN-caused SMA, but it's just a different gene that is causing this disease. And we have a very strong team that has been working on this gene therapy for the last 2 years, and we hope that we will be able to provide this additional therapy for another subpopulation of SMA patients in the near future as things develop through the preclinical phases here. All right. I think I'm going to just stop here. The preclinical data is really exciting, but I want to leave space for questions as well. And you know, thank you very much for your attention, and thank you very much for the MDA. They actually did sponsor me when I was a postdoc back in the day, and it was extremely helpful for my career, and of course all the patient foundations that helped sponsor these research studies that I've been talking about. Thank you for your attention.
Thank you very much for your update on clinical trials, considerations, challenges, and concerns within research, Dr. Meyer. We really appreciate you being here. We will proceed. It looks like we do have a few minutes for Q&A. I do see a question that came through. First of all, thank you for all the work you do to find a cure for these neuromuscular diseases. I'm interested in knowing if there's any research being done on regenerating the muscle by using family members' healthy cells.
By using family members' healthy cells?
Yes.
Okay. To the best of my knowledge, no. I'm not aware of any attempts to implant family members' cells into patients. I think one of the aspects there would be very similar to immunotherapies. We would have to be still very careful to evaluate whether the cells are actually compatible or whether there is any signals that could still lead to a rejection. But it's definitely something that could be considered. Yeah.
Thank you. That is the only question we have so far. We'll just give it one more minute here.
It was a lot of information, so maybe too much.
All right, I think we can wrap this one up. Thank you so much again for joining us today, and your knowledge and expertise is so appreciated. So thank you, and have a wonderful day.